All right. So what I need you guys to do now is we're going to do a little bit of practice with answering questions for osmosis. And this is called tonicity practice. So I'm giving you an example of a type of question that I would give you, and you're going to work on these in groups in a little bit on a test that have to do with using hypertonic, using isotonic, using hypotonic to answer questions. So you're going to need to copy these into a notebook. So make sure you have one out. If you don't have one out, pause the video right now, take it out, answer your questions. So when you come across these questions, the most helpful thing that I can suggest, and I say this every year to my students, and nobody ever wants to do it, but then they do it and they're like, yeah, that helped, is to draw a diagram. So when I give you a situation like this one, a red blood cell is introduced to an isotonic solution. So there's a little bit of content in here. You have to think in your head, okay, isotonic. You have to know what it means when you see the word hypotonic, hypertonic, and isotonic. So I know isotonic means that I have the same concentration of solute. So the way that I show that is I draw a little box to represent my diagram, and then inside my box I draw a circle. And I drew it over here for you guys already. So my yellow circle right here represents my cell, and the box around it represents the white here is the solution that my cell is in. Now, to represent the solute, or the stuff that's dissolved in my solution, I used purple triangles. So the way that I represent isotonic, meaning there's the same amount, is I put the same number of purple triangles on the outside and on the inside of my cell. And this shows me that they have the same amount. Now, water is always going to move in the direction of higher solute concentration. It's going to move towards where there's more stuff dissolved in the water. Now here, there is no more stuff dissolved anywhere. So what I would draw in my diagram is I draw one arrow showing water flowing out, and I draw exactly the same size arrow, try as best you can to do this, of water flowing in. Because this is how in a diagram we can sort of represent what we call no net movement, or no total movement in one direction or the other. So yeah, water still leaves the cell, but as water's leaving the cell, we also have water entering the cell as well. So it stays right around the same amount. It's at an equilibrium. And then what I would write, so there's my diagram, and describe what happens to the cell. Here's my description. So there would be no effect on the cell size since there was no net movement of water, no total movement, when a cell is in an isotonic solution. And this will be true whenever a cell encounters an isotonic solution, so where there's the same concentration on both sides. So sometimes I'll just give you isotonic, but other times I'm going to give you a situation. So here is another example of a type of question you might see about this. A cell with a 0.25% NaCl is put into a solution of 2.6% NaCl. So first of all, don't get holed up on the names of the chemicals. Just think of it as stuff, solute. So there's 0.25% stuff on the inside, and there's 2.6% on the outside. Now we know NaCl is salt, so it's just telling us how much salt is inside and outside the cell. So the way I diagram that, again, I draw my box to represent my solution, draw my circle to represent my cell on the inside, and now because it's only 0.25% on the inside, so I draw a few, and I'm using, in this case, my little orange, hexagons to represent my salt. So I put a few of them on the inside, but then I put a lot more, and I always exaggerate it just to make your life easier, on the outside. So don't worry about these numbers at all. Just look at these numbers and say, okay, which one's bigger? 2.6 is bigger. So 2.6 represents what's in my solution, so the solution has more stuff, it has more solute in it. So now once I have this drawn, now I think about my arrows. So I know that water moves in the direction of higher solute concentration. So it's going to move in the direction of whatever there's more stuff dissolved in the water. When I look at this, I see I have more stuff on the outside of my cell. So I draw a big arrow showing there's a lot of water movement out of the cell because it's going to head in the direction of higher concentration. Now I'm still going to have a tiny bit of water that moves the other way because water is always moving back and forth across the cell membrane. But when I look at my net movement, so the total direction this is going, I notice I've got a lot more water moving out than I do have moving in. So in my description, I write, the cell will shrink because when a cell is in a hypertonic solution, it shrinks as water moves towards the area of higher solute concentration. So remember, I got a lot of water moving out to sort of dilute the amount of solute that's in my solution, so that's surrounding the cell. Only a tiny little bit of water coming in, so my cell is actually going to get smaller. It's going to shrink. So that's how I want you guys to answer these questions. All right, so issues with tonicity. So here are some red blood cells. So if we were to put a red blood cell in a hypertonic solution like we just diagrammed, we know that hypertonic means there's more solute on the outside. water leaves the red blood cell, your poor red blood cell, it gets all shrunken, it gets disfigured. If we put it in an isotonic solution, look at these happy red blood cells. So isotonic, same solute concentration inside and outside. So water moves in at the same rate that it moves out. Everybody's happy. If we put red blood cells in a hypotonic solution, what we would find is that now all of a sudden there's more solute, more stuff on the inside of our cells. So water moves in the direction of higher solute concentration. So water is going to move into our cells. And those cells are going to puff up, puff up, puff up. And if we get to a point where they can't hold any more water, boom, they might burst or lyse. Fun thing about lysing, so cells bursting. In animal cells, cells burst. They can actually lyse, like we just saw with the red blood cells. But your plant and bacterial cells have cell walls, meaning they do not burst because they have this really thick carbohydrate structure on the outside that doesn't burst. But they can shrink. So plants actually, part of how they give themselves structures, they actually kind of overinflate. So they'll actually take on more water in their vacuole, and that'll give them some additional structure. So they don't burst, but they do shrink. And we have a special name for this shrinking. So if you take a look right here, you have, so here's my plant cell. So a hypotonic solution. So an animal cell, this would lyse. So in this case, we would have an animal cell. If we put too much water in here, it bursts. But this is a turgid, and that's the word for this, turgid plant cell. So remember, plant cells overfill themselves with water, so they put extra water in their vacuoles to give themselves a little more structure. So this is where a plant cell is happy. Your animal cell is very unhappy in hypotonic solutions. Your animal cell, very happy in isotonic, but your plant cell, not so happy in isotonic. This plant right here doesn't have as much water as it would like to. So see how the water pressure isn't pushing out on the cell wall, giving it some structure? This is what a plant looks like when it's wilting. So your plant's going to start to kind of lose some of its rigidity. It's going to wilt a little bit. And then here is a very, very unhappy animal cell and plant cell all the way over under hypertonic solution. So we notice that our animal cell shrivels, and if it's in a hypertonic because water's moving out. But we also notice that when water leaves our plant cells, what we see here is that the cell membrane actually peels away from the cell wall and it sucks in. And this is what we call plasmolysis, or when the cell is plasmolysed. So much water is left to our plant cell that that cell membrane pulls away, shrinks, and causes our plant cell to kind of cave in on itself. All right, last one. So facilitated diffusion. So this is for molecules that are too big to pass through the cell membrane. So what they have is they need a little bit of help, and that's what facilitated means. Facilitated means with some help. So it's still diffusion, which means it still does not require energy, and here's where your notes go. So since they are too big to go right through the phospholipids, they can't squeeze through the phospholipids, they need a protein channel that lets them pass through. So it's basically a tunnel for them to squeeze through. There's still no energy required, and it still moves from areas of high concentration to low concentration. So again, no energy needed, just squeezing through a channel. So over here on the left-hand side, we have our diagrams of our two types of passive transport. There's simple diffusion, which is shown by the purple circles that are tiny nonpolar molecules. They just squeeze right through those phospholipids. And then over here, we see our facilitated diffusion. So these are bigger molecules like our red triangles right here. So they're going to use the channel. So it's just a tunnel that it uses to pass through the phospholipids. And then like our ions over here, or blue circles, they can also use a channel to move through. All right. So what we're going to do now is pause here. You guys are going to do a little bit of review working on passive transport. If you have any questions, please feel free to let me know. And you should have your notes filled up through facilitated diffusion. So you should be on the part of active transport.